These are going to be your DNA notes, which are part of the DNA RNA unit that we're doing. So it's going to talk about these notes will talk about the DNA structure and DNA replication. So first of all, the DNA structure. So I'm hoping that you know that DNA stands for deoxyribonucleic acid. Now think of DNA as a ladder. So the rails are sugar and phosphate, and by rails I'm referring to this part here. And they alternate. So we'll look at pictures in class that show how they're actually combined, but they're going to alternate sugar, phosphate all the way down the backbone. And then the actual steps that you see in the middle are base pairs. So if you look at this color over here, it shows yellow is A, green is T, blue is C, and the pinkish red is G. So something very, very important that you need to know is that A pairs with T, G pairs with C. That's something you just have to memorize. You need to know what that is. And then all combined, they make something called a nucleotide. So that is a sugar, a phosphate, and a step, or a base pair, makes a nucleotide. And then I have that written down there for you as well. So back to the bases. So they are two different types based on structure. So you have something called a purine and then a pyrimidine. So a purine has a double ring, while a pyrimidine has a single ring. The easiest way I think to remember this is thinking about the bases. So the examples, you have cytosine, which has a Y, and thymine, which has a Y, and both of those are pyrimidines, which also has a Y. The purines then are adenine and guanine, and neither one of those have a Y in them. So purines are double ringed. Think it's a shorter word, but it's double. Pyrimidine's a longer word, but it's single. You just have to come up with a way to remember this. So then base pairings. So we talked about how A pairs with T, G pairs with C, but why do they only pair certain ways? Well, it's called base pairing, and the reason for that is that purine and pyrimidine makes the base pairing. So you have to have a purine and a pyrimidine to make a base pair because of how the bonding works. And we're not going to get into all the bonding that goes on, but if you ever take AP Bio or even chemistry, you'll talk about it a little bit. So a purine, we said, was adenine, guanine, because they don't have the Y's, pyrimidine, cytosine, and thymine. And if you notice, these two pair and these two pair. So we have a purine and a pyrimidine. And something very important you need to know, the hydrogen bonds are what holds the DNA together. If you remember, we talked about how hydrogen bonds are weak bonds. So because the bonds are weak, that's why it's so easy for DNA to unzip when it replicates. So just think about the hydrogen bonds hold together your A and T or G and C. So then quick review of how DNA is formed, and I'll show you guys a picture of this in class. So you start with DNA. It wraps around histones. So that's written right here. The histones coil into nucleosomes, and then the nucleosomes coil to form chromatin. Now these words should be familiar. Chromatin into chromosomes, which we should hopefully remember is made of two chromatids. I'm not going to be able to spell the whole word on there. Two chromatids make a chromosome. So it starts as DNA. So chromosomes are made of very tightly coiled DNA. So DNA replication then. This is simple enough. DNA to DNA. You need to know that replication is DNA to DNA. It's making a copy, an exact copy of something. So why does DNA even replicate? Well, when a cell divides during mitosis, which we also just finished talking about, each cell has to get equal amounts of DNA. So when does this replication occur? Hopefully you remember this too. The S phase of interphase is when DNA replication occurs. So as a result, you get two identical complementary strands, and they follow base pairing rules, which means A and T, G and C. That's what I mean by base pairing. So how does DNA replicate? Well, each strand has all the information it needs, but it replicates through a 4-part process, but that's just for eukaryotes, okay? Eukaryotes is more of this 4-part process. Whereas prokaryotes, it's a single fork and it just kind of goes, although they still kind of do the parts, but we're only going to talk about these 4 parts in terms of, like I just said, the eukaryotes. But you do need to know that in prokaryotes, it begins at a single fork and continues in both directions. This is like, because DNA, it's not exactly in a straight line like that, it's usually coiled up inside, but it'll start at one single point and it will go all the way around until the whole strand has been replicated. Eukaryotes, let's say they have the same kind of circle. They don't, but let's pretend. It would start here, 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 and hundreds of other places and go, because eukaryotes have much more DNA than a prokaryote. So eukaryotes have to start this replication in more than one place. All right, so the first step of DNA replication then is initiation. So big picture, you have an initiator protein and it binds to the origin of replication. This origin, that's a word that you definitely need to know. The origin is the start point. So the initiator protein binds to the starting point, and that's going to start off our process of replication. So after initiation comes unwinding. So DNA is in that double helix. You can't replicate when it's wound up like that. So it has to be unwound. So DNA is held apart by these single-stranded binding proteins. And then a helicase is what unwinds it or unzips it. So it breaks, like I said earlier, the hydrogen bonds. They're weak bonds. They're easy to break. So the DNA helicase is going to break those bonds, but very important, those single-stranded binding proteins have to hold the strands open here and here to make sure that the helicase can function. So as it unwinds, there are 2 strands. So we have something called the lagging strand and the leading strand. Basically, big picture here, the leading strand is a continuous replication. It's one continuous part. It doesn't have to stop and start, stop and start. The lagging strand has to stop and start. So it produces something called Okazaki fragments. And don't worry, we'll discuss this. I'll show you some pictures. But it's these fragments that have to be later put together. So lagging is discontinuous. Leading is continuous. Think about it in terms of a race. The leader is going to be the one that wins, whereas the lagger is going to be the one that loses, okay, because they're slower. So the lagging strand is slower than the leading strand. And then you may have noticed here this 3', 5', 5', 3'. We'll go over that more, but it's just the direction. So the ends are either 3' or 5', and they always go 3 to 5 or 5 to 3. So there won't be any other numbers you'll see. Then the elongation happens. So if you're elongating something, you're making it longer. So what happens is the DNA, as it starts to replicate, it's becoming longer and longer. So this is the stage where the DNA is actually replicated. So we started it at the origin or the start site, and then we unzipped it. But now we have to actually make the copy. So it always moves from 5 to 3. This will be a quiz question. This will be a test question. This will probably be an EOCT question. You need to know that replication always goes from 5' to 3'. And it starts at something called the replication fork. So something called DNA primase. So a primer, if you put on like a paint primer, it's first. So think about primase goes first on the lagging strand. It adds a short segment of RNA, which is an RNA primer, onto the lagging DNA strand. Then, I know this is confusing because this is RNA, but from there, the DNA will replicate. Okay, The DNA polymerase is what adds free nucleotides to the chain. And this is also what creates those Okazaki fragments. Now, polymerase, you can think about it poly, if you remember, means many. So think about it as polymerase is the time when these many nucleotides are added. So if there's a T, it's going to add an A. If there's a G, it's going to add a C, and vice versa. So here's kind of a picture to show you the elongation, and if I were you, after I explain it, I'd pause it and look at it for yourself and see if you can understand it, and then you might even want to sketch it in your notes. So we have right here shows our direction of replication, so it's going this way. So your helicase, remember this unzips. Okay, then it shows your 5' and 3'. So if you remember, if we go back, it always moves 5' to 3'. So we're always going to go from 5 to 3. So the blue strand is our something called our template DNA. So it's what you make the one off of. So if you notice, the blue strand starts at 5 here, and the 3 is down here. So because it's going 5 to 3, they have to go opposite. So the strand that's being copied has to go 5 to 3, but it has to be starting at 5 up here and ending at 3. Think about it like magnets. You can't put two plus sides of magnets together and two minus sides because they repel each other. Same thing. If this was a 5 down here, it wouldn't work because they'd repel. So you need the opposite. And so these, as you notice, are your fragments because it goes 5 to 3, and then it has to start where the next DNA polymerase was and go 5 to 3, 5 to 3. And then if you notice, this one also has this DNA ligase, which links the strands. I know these are a lot of terms. We'll do a sheet to fill out kind of structure function of each of these things that are used. And then this way, this is your leading strand or your winner. The lagging is your loser. So the leading is going quick because 3 is at this end. So the 5 gets to start here with the red, and it goes all the way down. It doesn't have to stop until it gets to the end. And then just another picture showing you how this works. So you have one going this way and one going this way. So they're always going to go opposite ways. And then the final step, termination. So how does the DNA know when to stop? So in some DNA, when the two forks meet, it's going to stop. Okay, and this is especially in a prokaryote where when it reaches where the other fork started, it's going to stop because it's done. But in others, there's a specific termination sequence on the DNA. And that's something that we'll get into once we do the next video with RNA. We'll talk about the termination sequences, but just know that sometimes there's an actual sequence that says, okay, stop. And then how is it rejoined? So like I mentioned in the last picture, the fragments must be joined together. Something called DNA ligase is what links those fragments together, and it forms the full complementary strand. And then just another quick summary of replication. So this is the overall direction, if you notice down here, is moving this way. The direction is going to move in the way that it's unzipping. And then here's your helicase, here's your replication fork. You see all these different things. So your primer, your fragment, your polymerase, your DNA polymerase, your ligase, all of those important things are all on this picture. And then just a quick review. So we have initiation, it starts, elongation is when it's forming, and then termination. And then there are different models of replication. So there's 3 total. You have conservative, dispersive, and semi-conservative. Semi-conservative is what scientists follow. So it means each strand of DNA, so remember DNA is double-stranded, so there's 2 strands, serves as a template. So a template strand is what you're making a copy of. So this is shown here in this picture right there. Okay, so conservative doesn't really make sense, so we don't like that one. Dispersive is even more confusing. They don't like that one either. They like the semi-conservative. So if you see the original one is dark blue, so one dark blue goes here, one dark blue goes there, and then the light blue is the copy. So then when it further replicates, the dark blue makes a light blue copy, but then the light blue makes another light blue copy of itself. So the colors are just kind of showing you how this functions. And there can also be replication errors. So their error rate is 1 mistake per billion nucleotides. That's a pretty low error rate, but how they're caught. So nucleotide selection. So the accuracy rate is very high. And once again, nucleotides, this is pairing A with T, G with C. Proofreading. So your DNA polymerase is also a good proofreader. If a base is added, it will stop, remove the incorrect base, and replace it, then continue replication. And as a final last plan, if 1 and 2, neither one work, you have actual mismatch repair enzymes that go find the problem and replace the base. So it's very, very uncommon to have a mutation that happens during this replication. And then just some replication practice. This is something that we will do in class, a lot of actually, but we're just going to practice giving the complementary strand. So C pairs with G, C pairs with G, G pairs with C, T pairs with A, T pairs with A pairs with T, T pairs with A, G pairs with C. And then think about what numbers. So I said about magnets and how they repel each other if they're like ones together. So because there's a 3 here, we're going to put a 5 prime. Because there's a 5 here, we're going to put a 3 prime. So let's do the next one. So T goes with A goes with T, G goes with C, G goes with C, C goes with G, T goes with A goes with T, A goes with T. And once again, 5 prime at this end, 3 prime at this end. And the last one, so A goes with T, G goes with C, C goes with G, C goes with G, C goes with G, T goes with A goes with T, and G goes with C. And 5 prime, 3 prime. And that is it for your notes for this one.